Amino acids a lot of times are referred to as the building blocks of proteins. And what this means is that amino acids are going to really be the monomers that we use to make up proteins, which will be much larger molecules, which will be polymers. So the proteins are made up by hooking together a whole bunch of amino acids in a row. And usually we're talking somewhere on the order of a couple hundred up to maybe a couple thousand amino acids joined together. So first here, we want to be able to describe the structure of an amino acid. So as we begin to do that, there are, if we just break down the word amino acids, there's two functional groups that we're always going to find in amino acids. So the first functional group comes from the first word in the name, and that's the amino part. So we will always have an amino functional group. And just to remind us all, the amino has an NH2. So we're always going to have one of those. And then the other one that we're always going to have comes from the second part, which is the acids part. And carboxyl groups a lot of times are thought of as acidic functional groups. So carboxyl groups, recall, they're going to be like this. Okay, so we're always going to have those two kind of defining functional groups when we do start talking about the overall structure of an amino acid. So to get us started with the amino acid structure, right in the center of our amino acid, we're going to have this carbon, which we refer to as the alpha carbon. So I've drawn the Greek symbol alpha right there next to the carbon to indicate that that's my alpha carbon. Now, we're also going to have, just as we mentioned, we're going to have to one side, we're going to have the amino group. So I'm just going to draw it backwards here since it is pointing in that direction. And then to the other side, we're going to have that carboxyl group that we mentioned earlier. So right away, you can see the two different groups that we're going to have on this molecule. So we've got that central alpha carbon. We've got the amino group to one side. We've got the carboxyl group to the other side. We're going to have a hydrogen attached. So I'm just going to point that down for now. And then up above, I'm going to give it its own color here. So put this one in red. This is going to be what we often refer to as the R group. So that portion right there is highly variable. And that's what's going to give the different amino acids their unique functions. So our cells make their proteins from 20 amino acids. And those 20 amino acids all have this structure essentially the same. But the R portion is going to vary quite a bit. So if we look at that, this image shows us the 20 different amino acids. And you can see their R groups are being shaded in different colors on this image. So all of the portions that are shaded, those would be what's considered the R group. Sometimes that's also referred to as the side chain of the amino acid. And those side chains can have a lot of different properties depending on their overall configuration and the type of bonds that they contain. So notice that the first row is going to all have nonpolar side chains. So we tend to call those our nonpolar amino acids. The second row is showing us the polar ones. So they have quite a few polar covalent bonds in their side chains. And then the ones at the bottom actually have some charges in their side chains, and that would be due to the fact that they do contain some functional groups like amino groups and carboxyl groups that are going to give them those charges. So when we look at an amino acid, we want to keep in mind the part that's always going to stay the same, because that's going to help us to identify all those 20 different ones, even though in this course we're not memorizing what each side chain looks like. So again, when we're trying to figure out and really define our amino acids, we're always going to look for the alpha carbon. We're going to look for an amino group. And here we're going to look for the carboxyl group. And if you can find all of that coming off, you can probably find your amino acids. Now, as we mentioned, these amino acids are going to be used to make proteins. So they are going to get hooked together to make larger molecules or larger polymers. Here I have two different amino acids drawn. These are just representing generic amino acids, which is why I haven't really put any detail on the R group or the side chain. So this would apply to any two amino acids. We could take them and they could be joined together by a dehydration reaction. So with a dehydration reaction, hopefully you can recall that what that does is it is going to remove a water molecule. And when it does that, it joins the monomers together. So that will be the case here. So we were actually going to be removing the OH from the carboxyl group of one amino acid, and we will be removing one of the hydrogens off of the amino group of the next amino acid. So once that happens, we're going to have water come out. So I'm going to put my plus, my H2O over here because that will be generated in all of this. And then if I draw the result, when I hook those two amino acids together, take me just a moment to draw it here. And I'm adding in those alphas, hopefully, so you can track the centers of each of the amino acids that we had previously. Draw those R groups back in here because they are definitely there. Nothing happened to them at all. So this is the end result that we're looking at. Now, where we lost the water molecule is right here. That's where we just formed this new bond. That connection right there, it does have a special name for it. That is what we refer to as a peptide bond. So the peptide bond is the bond that gets formed between amino acids when they are joined together by a dehydration reaction. That peptide bond is going to form between what was the carboxyl group of one and the amino group of the next one. And this end result here, since we only hooked two together, this would be what we would call a dipeptide. But usually we're going to have many more joined together. Usually we're looking at several hundred to several thousand of these joined together. And that would just be the same reaction over and over and over again. Now, if we just look for a moment at this molecule that we have constructed, it does have two kind of unique ends to it. This end over here, notice it has an amino group on it. That is what we tend to refer to as the N-terminus. And then on this end, we have a carboxyl group, and that's what we tend to refer to as the C-terminus. These are just names to refer to those two different ends of the molecule. Regardless of how long this molecule actually ends up, even if it is 1,000 amino acids long, we're still going to have one end that has an amino group and one end that has a carboxyl group on it. Also, just emphasize again here, the R groups, they do not participate whatsoever in the linking together of the individual amino acids.